Hello everyone and welcome to our NGS-CN Summer School online collection on spatial transcriptomics. Before we jump to our next speaker, let's take a look once more who we are. So it's a next generation sequencing competence network that consists of 4 production sites as you see on the map. So in 2019, DFG has funded centralized sequencing applications. There were quite a lot of calls, and within each call there were a lot of projects. And from 2024, DFG has changed the strategy towards individual sequencing applications. That means that now there is no special deadline for a centralized call, and you can co-apply at the DFG together with the chosen genomic facility. If you wish, here is a QR code, contact us, and then we could take a look and maybe sequence together. If you want to know something more about our news and upcoming events, then you also can take a look at the QR code, scan it, and get into our mailing list. Let's grow community together. And with that to be said, let's welcome our next speaker.
My focus of my PhD is basically the ring versuch, it's called, and it's a collaboration of different centers, and we kind of do things together, and then we see if we can get some single cell data or atlas out of it. Today, I would just discuss the data integration strategies in this multi-center RNA sec. Yeah, so basically now, given that we have all these high throughput single cell sequencing data available, people are starting to generate what we call the cell atlas. So basically, it's a catalog of all the different types of cells based on transcriptomic diversity belonging to a particular organ or cell or at a different levels of resolution. So eventually, what we would definitely like to do as a field is to make a comprehensive universal cell atlas. And now that's a grand vision, basically trying to cover every kind of cell and diversity that exists in the human body, for example. For this to happen and for meaningful inferences to be drawn out of these atlases, we need to have population scale diversity. So basically a lot of individuals and not just from one particular demographic, but many different demographics. Secondly, we would also want to have as many organs and as many sub areas that we can actually sequence. And then, of course, we want all of this at a single cell resolution. So current approach to this aim, this big grand aim, is multifold, and I could break it down into the scope of the study and then the strategy employed by different studies. And in the beginning, we had this human cell atlas approach, which basically was a decentralized way. Different labs with different expertise produced different data, which then was put together to start to construct what the human cell atlas would be. However, that did come up with its own problems, and subsequent studies actually changed from multi-organs to organ-wide studies or to centralized sampling. So basically at one particular single cell center, you had the sequencing, and then you construct this atlas. And so that's the approach which has been happening in the last time. And the problem with this approach, of course, is that we cannot have population level diversity. One center cannot, so sitting in Germany or in America or wherever, you cannot cover the entire human population. You basically need to have a decentralized approach to this thing. And so centralized approaches are not scaled for population level diversity. We need decentralized approaches. And then, of course, decentralized approaches, as was witnessed then in the human cell atlas, is that they have problems of data compatibility and data integration. So something that Katrin also already showed you, that you have quality differences between data and then can lead to batch effects. And so having a decentralized seems like a grand idea, but then in effect, it didn't really work out that well. So most studies, again, as I said, look for a very centralized approach, or a single center doing all the time. Another approach of the scope of the project is another thing, which is basically people try to, again, do grand projects with multi-organs or like a whole lung atlas, and this what it actually does eventually is that it lacks sufficient resolution, which we actually want to leverage later on, which is at the single cell level, and we want to have very high resolution in our data. And because of the bigger scope of the thing, we don't have we can't discuss all the finer diversity. And then all these grand atlases then, of course, lack annotation or the biological context in which this diversity was discovered. And as an example, I just want to show, and it's no way in trying to put down this study, but this is the human brain atlas we have right now. This is the state-of-the-art human brain atlas, and published in 23. And what you can see is that given this grand, again, broad scope of trying to elucidate all the cell neurons of the brain, what we end up with something like called splatter neurons. Yeah. So that's just a symptomatic, symptom of what happens when you try to put very diverse cells into one, and then you end up with something like a splatter, and then you end up teasing apart all of these. And again, splatter cells then is symptomatic of the idea that we should be doing more regional and content analysis. So basically, we need decentralized, community-driven tissue region-specific studies, and we need to have different labs working with each other. So that's the basic idea. And of course, we need to be focused on certain tissue regions. And I mean, again, in 2023, there was a Nature editorial saying something that we had already started working on, which is basically that mega brain project teams need to be talking to each other. So basically, this idea of decentralized community-driven atlases. Now, of course, as I already said, we face a problem when we have these decentralized data. Every lab then produces its own data, and then you don't know, you have this typical 5 blind men touching an elephant problem, identifying it as a snake or as different animals. And that's what happens if we have different labs working and have data which is produced, which is not compatible. Yeah. And specific to then our field, and also to our question at hand, then you have, of course, inter-laboratory procedural differences. So everybody has different machines, different chemicals, or different just manual labor can have differences. And then, of course, we have the problem of the data, which the type of data we are dealing with. And if we are talking about single cell, the sparsity is a big problem. There's stochasticity, that means which genes are covered, not always, and uniformly covered across all cell types and all technologies and platforms. Then you have different protocols which are used sometimes to enrich for a certain type of cells, and data can be generated using different enrichment protocols, which might not be compatible then. And then, of course, there's annotation, gene annotation. This is probably like something that you face when you deal with this data, basically that there are different versions of gene models which people use sometimes, and this can create data matrix problems. And of course, data quality research. We have this challenge, and I would like to discuss how we are going to approach this challenge. So the results that I'm trying to present are in a context of asking the right questions, or then more than just like selling the results, let's say. It's about identifying what are the sources of biological and technical variation, because we want to do something called batch correction. So these batch effects that we have in our data, assimilating data from different sources, needs to be accounted for. And the first step is to understand that there's always a conflation of technical and biological variation in single cell data, and we need to identify what is our batch so that we can account for it. And that's a very big question, because it's a very highly research question also, because it has an impact on the eventual cluster that you saw. So for example, you had these low quality clusters in the last slides which was related to low mitochondria, etc. So all these batch effects had to be accounted for. And then, of course, once you've identified the batch factor, another intermediate question would be, do I even need batch correction? Do I even have batch correction? And this you should find out while you're trying to discern the technological and biological variation in your data. And then, of course, finally, you have which data integration method should I use to integrate my data. Dive into the case study, which is the Ringversuch. It's called the Ringversuch because it's a round robin experiment where different centers and sites process similar or the same kind of replicates, and then we try to see if we can put this data together and how compatible this data is. Ringversuch is the German word for round robin experiment, and that's how this experiment was planned. So in the basic concept of it, we have in the beginning we have 3 human control subjects, and all 3 are females. And then we have the focus of our study is the striatal region, which is basically localized to the dorsal striatum here, which is caudate nucleus and putamen. These are 2 parts of the striatum. And then what we basically do is we take different tissue samples, and then we sent it to different centers across Germany, who all followed the same kind of standardized protocol according to a SOP, and did these things independently in that sense, and then sequenced these data and then sent it to the center to Bonn, where we processed the data. An important part of this experiment was that basically, again, we are talking about the brain here, and we have our focus was neurons. As some of you might know, that the two basic classes of neurons of cells in the brain would be then the neurons and the glia, neuroglia, and we were actually focused on the neurons. And there we used a neuron antibody, and we used two different. So besides having this distributed sampling, we also try to see, okay, what is the impact of different sequencing platforms when we use the same kind of data and generate the same kind of data on different sequencing platforms. And here we use a microfluidics base, which is basically 10x, and then the nanowells system based on BD Rhapsody. And then this is generally the sample design. Each site has basically a tissue coded nucleus and a Guttman sample, and certain sites for to be specific had also BD in addition to 10x. Sounds like a very fast introduction to this, has the key message that this is in a way a very complicated design, but also a very thorough design to be able to tease apart what are the factors, biological and technical, which contribute to the variation within single cell data. And here we have 3 subjects, 2 tissue regions, 7 sites, 2 platforms. So if you just do the combinatorials, you can see that there are so many axes of variation inside this data, and one needs to kind of tease them apart to be able to discern biological and technical. And so to kind of summarize that, as you can see, you can have inter-site variation inside it. And this is a variation that you don't want. You want basically different centers to produce the same kind of data that you can easily integrate it. So this would be a technical variation that you don't want in your data. Inter-subject. Now I've marked this as technical and biological, and maybe it will be clear more later. But in general, you would assume that biological variation should be, inter-subject variation should be biological. But given that we want to make a cell atlas here, we are currently not interested in, and for a specific organ, we are not interested in subject variation. So this is a nuisance factor for us. So we have only 3 subjects, and we're interested in the intercellular variation, which is the biological question that we're interested in. And of course, there's protocol, data quality, which are again technical variations, and then interplatform, which is the 10x and BD problem. And then there's the intertissue, which we also want from, for example, what is the variation in the cellular composition, signatures of both corticulus and putamen. Diving straight away into the results, one way of looking at the data, how this variation is shaping our data. Yeah, so this is basically the results of the single cell data. And here on the left-hand side, we have 3 different metrics which go from left to right. We have the UMIs per nucleus, then we have the number of genes per nucleus, and you have the mitochondrial genes per nucleus. What we see is that in the platform variation, so the technical variations according to platform, there's a clear bimodal distribution of the quality metrics across all 3 metrics. So the number of UMIs is less in BD, the number of genes per nucleus is less in BD Rhapsody, and the mitochondrial content of the cells in the nuclei and the BD Rhapsody is also higher. So overall, the quality differs between the platform. So when we are doing multi-platform studies, we can clearly see that that is one of the major factors of technical variation. And when we move to subjects, we basically see that there's generally a very unimodal distribution of UMI and genes, but we have a certain kind of differences in the mitochondrial content, and that is for one subject, for example. And this kind of also highlights that subject-specific or sample-specific variations can be important and can be a batch effect in your data, and they are conflated with then technical variation. And then if we go to the sites, we actually see that 5 out of the 7 sites have a very homogeneous or very congruent data quality, which is quite surprising given that they are all, given that we expect in general different sites to produce different kinds of in terms of the technical aspect. except we have some outliers, which is green, basically the bond in both in the UMI and the number of genes. And we have an outlier in terms of the red in the mitochondrial percentage. And as we see, we can compare it to the above one, which is the subject. And you see that since this site only produced only sequence subject from this subject, the site and subject variation then therefore is conflated. And this is mostly explainable given that overall the sites showed very homogeneous distribution, that you can kind of say that it's a sample or subject-based variation. So basic message of this is that if you follow the protocol to the T, let's say, you have a very similar data set. So in general, when you're trying to combine data from different centers or sites, if they follow the same standardized protocol, you actually get a very good congruent data. Of course, we saw that there's bond, and then one asks, okay, so why did bond have this lower UMI and genes per nucleus? When we look at the cellular composition of the quality metrics, what we see basically is that these quality metrics are driven largely by the cell types. So basically, we also know post hoc and in general, this is now becoming a common knowledge in the field that basically the glial cells have a lower RNA content, or the transcription rates are smaller. The transcriptomic content is smaller than the neuronal content, and this is reflected in our data. So basically, bond having lower quality metrics for UMI and genes is directly related to the kind of cells that are comprising the samples. Yeah. So again, we can see that site per se is not the problem here. We have other factors again which conflate biological variation. So you would imagine that intercellular variation should be a biological aspect, but here in the brain, this can also be a technical variation. Yeah? Okay, so yeah, and so this basically helps us to identify that, okay, if there's a protocol breach somewhere, or basically something that didn't work out, and we found out that the neuron and antibody used in neural enrichment in bond was a bit different from the rest of them, and that kind of caused this. But overall, the message is still the same. Now we say, okay, so this is quality metrics. Now, what about the transcriptomic fidelity? So how do I measure the transcriptomic fidelity between different sites? So for this, what we did basically is that took each and every took individual samples, or we kind of integrated them using SCVI, which is unsupervised. So basically, we project them to a common space, and then we label clusters. We need these label clusters to judge how predictable is one sample based on training for another sample. So, so once we have these common clusters, we know, okay, then we can go for the judgment for testing whether a particular sample can predict the cell clusters of another one. So here, what we did then in the second step is that we train the model based on the clusters that we identified in the global integration, and then we do this on a single sample level. Now, after training the model on a single sample, we try to take the other samples which belong to the same subject and tissue, and then see, okay, can we predict the cell types of those just based on this one single sample? And then when we compare the ground truth, what we call the common clusters in the first panel, with the ones which are predicted, and if they are the same, let's say they are called then 100% accuracy. And so we did this analysis in such a way that the other confounders in our data, basically the inter-subject variation or inter-tissue variation, was not included. And what we see as a result in terms of the accuracy of prediction is that it's very high. So if you took one sample and you compare it to another one and look at the transcriptomic signatures, one could say, yeah, you could. The data is very, very compatible, and we had accuracies over 90 more or less across all comparisons that we made in terms of, and basically, again, it kind of emphasizes that inter-site variation is not the problem, and now in two different levels. So one level is, of course, the quality control, and now the second level is here, the transcriptomics. So this is the unit, so the clustering, this is the unit which we try to make cell atlases on. Yeah? Finally, then we also wanted to see, okay, we've now established that site is not the problem. Then where does this variation come from in the transcriptomic space? Again, we did a similar analysis to the last one, but this time using PCA space as our dimensionality reduction method, and we could calculate what is the contribution of what is the distance between what is the inter-subject variation, what is the inter-tissue variation, and inter-site variation. And interestingly, inter-subject variation appears to be the most discrepant here than inter-tissue variation. So we have more biological signal than technical signal in our data, and that's actually a big confidence booster for doing multi-center studies, which are this chunk. Okay. So of course, now we've established that the data is congruent between the different sites. The highest contributor of variation is subject, and then tissue, which is a biological factor, which we actually are interested in. And we want to now see how can we then accommodate this batch effect, or what is the batch effect here that we need to accommodate. So we look at the data in in a common space, common dimensionality reduction space. This is a UMAP. Sorry for missing the axes. And if you color the different clusters or different cells inside the UMAP by different categories of variables that we have, for example, cell class, subject, tissue, site, sample. I think it's a lot to kind of grasp in each and every aspects, but the important parts over here are, if you look at the site part, and if you compare it to the subject part, what you realize here very clearly is that what we saw in the last graph, that the cells from the same subject kind of come together, irrespective of which site they belong to. Yeah. And the tissue has a slight, within each subject, there's a slight variation within the tissue, but this variation within the tissue between CN and PU, for example, within one subject is much less than any other axis of variation. Another thing that we noticed from here is that this outlier site that we had, basically the Berlin site, which processed only one of these subjects, and the only site to process the subject, is also within the same subject, we have a very huge variation between two samples, so between CN and PU. Now, there are multiple reasons why this can happen, but as of now, that would basically mean that we need to account for this inter-sample variation to be able to integrate our data. And so basically, given that we had several lines of evidence telling us that site is not the discriminatory factor here in terms of technical variation, subject is definitely the most potent, but we also need to account for the highest inter-sample variation. Therefore, we used sample ID as a batch factor, and it of course includes different other levels of variation. So we use that. And then the idea is then, of course, how do you do this? Which method do you use for doing your batch integration? For this, we have there's a benchmarking study. There's a tool called SCIB. It's a very highly cited paper. Basically, with the group which actually devised this tool, we different integration methods to see which one actually scores the best. Now, the key takeaway from, instead of having to discuss all the details of what the results are, the key takeaways here are that the performance of these values usually use methods vary significantly. Yeah. So so saying that one method is the best, or this has always performed well, is not completely correct, but depends on your kind of data. And a classic example of this is when you look at the integration of Liga and the metrics. So these are methods lined by their performances. On the top, you have SCGen, and the bottom, you have Liga. And you see that it's even, the performance of Liga is even worse than unintegrated data. Now, this doesn't mean particularly that Liga is a really bad method for integration. What it clearly says that given the data at hand, this is not a good method for this data. So I think the key takeaway here is that you can benchmark it, and then you should see some metrics and guide your decision on which integration method to use based on these benchmarking, but also realize that choosing one method just randomly, which is popular, is not always the best choice in this case, and that choice has to be made based on your data. We chose Striatal here because the top 4 performing methods were all supervised. So basically, since we are constructing a de novo atlas where we don't know what the cellular diversity is, we chose a method which is not guided by already defined clusters, and therefore we chose Striatal V3. And when we try to integrate it, it looks pretty good, contrary to what it looked like pre-integration. We have a very good integration of subject, tissue, sites, and sample. Now, another thing that I wanted to show in this argument of on this thought process of integration is that, one, that we said that, okay, you have to choose methods based on our data. And another example of that is when you saw the 10x and BD differences. To integrate 10x and BD differences, our Striatal method didn't work. And we still had really like separate clusters for for 10x and BD, and we needed to use another method, so which had actually more than one batch variable permitted. And here we use harmony, and then we again got a different, very good integration eventually. But again, this was to kind of showcase that within a particular study, we had to use two different methods of integration, because depending on the batch factor, the variance in the data, and also how deep the contrast is between two batches, you will have to make decisions on which method to use. And similarly, things that I have not, then finally we go on further for other levels of integration where we map the data from other studies. And there we use, again, supervised methods, because that helps to accommodate more varied batch effects. And again, the point here being that depending on the task at hand, the question in hand, you need to choose the right method of integration. In summary, if somebody is looking for tips of how to do data integration, the important aspects over here is that, first of all, you need a very good interrogation of your data to see where the variation and what kind of variation is in your data, what contributes to this variation, and then define the question that you want to answer. So, for example, if you want to make a population level atlas where inter-subject variation would be interesting to us, then we could not call subject a technical variation, technical factor, or it could not be a part of the batch effect. Yeah? However, in this case, it was not the case. So we try to integrate over subjects also. The second thing is that so that's the important question, important point, that you need to define what is unwanted variation in your data. Yeah. So, for example, you have studies where you want to look at the development trajectory, and you want to then not. And then if you have cells in different phases or in different cell cycle things, you don't want to factor out as a batch effect, because that's the important question for your study. Then the other aspect is that the choice of integration methods can be guided by benchmarking and doing several studies. But again, there is subjective choices to be made. The contribution of the researcher, therefore, is very important in this case to make the decision. You have different guidelines and metrics which tell you, which can guide you to how you want to choose the method. But eventually, understanding the biology is very important for choosing the method, and therefore then determining how the results look like. As an overall message here, in terms of using decentralized, because this part of the presentation is just a part of a bigger study, I just wanted to make sure that it's understood in that context, that we wanted to have to kind of put through this message that decentralized atlases are possible. Intersite variation can be easily accounted for, and this is very dependent on what happens in the lab. Yeah. So a lot of things that which come up in the data can be dealt with bioinformatically, but if the underlying data is not good, there's no saving the data, let's say, or there's no bioinformatic method which can actually overcome bad quality data. And that's the underlining theme here. Thank you. That's it for now. I just want to acknowledge the lot of people. So Joanna is here in the audience, I saw. But there are also Andreas, hopefully, and there are Janine, and a lot of collaborators who have taken part in these 7 centers. They've done a lot of work. It takes a lot of effort to put all these things and coordinate the activity of 7 centers and the NGS-CN Center, and then this NGS Competence Network, therefore, has actually successfully be able to do such a complicated project. Then, of course, people at my group in DZNE in Limes who have also contributed, and most importantly, credit to my 2 professors, Professor Mark Bayer and Professor Schulze, and that they had to do that stuff. Thank you.